Okay, the secret of successful revision, the secondary edition. We're going to start off by briefly revisiting the first year's notes on revision. As mentioned before, pen and paper are often the best tool for helping you learn actively. We're all different. The advice there is fine for most people, maybe not everyone. If you have your own tried and true system for revision, that's fine, stick to it. For a bit of variety, you might want to try some of the things suggested here, but that really is up to you. Revision is a hard process for most people. Whatever approach you decide on, don't confuse being comfortable with being effective. Effective study is hard work, there's no kidding about that. We talked about active learning. This is more than just sitting back and letting the knowledge wash over you. Sadly, sometimes without leaving a trace. And here's the example we gave last year. And in my previous talk on this, I mentioned my old organic chemistry lecturer who used to say that education is that indefinable thing that passes from the notes of a lecturer to the notes of a student without going through the minds of either. Hopefully that's not going to be the case here. Writing's highly active, so is drawing, so is mind mapping, so is composing little ditties if that's what floats your boat. Whatever suits you best, I'd advise you trying a blended approach, try different techniques, for a little bit of variety, but also to find out what works best. Okay, so some general advice. Always worth reiterating, read the complete exam paper before starting. Find out how many questions you have to answer. This will be on the first page, so have a little read of that, and then look at the questions and think about which ones you're going to answer. Some of them will stand out. You say, I want to answer that. Some of them, I wouldn't touch that with a barge pole. And the temptation is always to do your best question first, which may be fine, but make sure you don't overdo it and spend an hour doing one question, leaving you with perhaps 40 or 45 minutes to complete the other three. And keep an eye on the clock, of course. Facts are important, but the second year we're looking more for understanding. We're looking for evidence that you understand the topic as a whole, and frankly you've read around the topic a bit as well. That is where you will pick up a lot of marks if you're a very good student. Common mistakes, answering the wrong question, which is always tragic. Sometimes we see some excellent answers for the wrong question. Very little we can do about that. The other thing we see very often is taking too much time over relatively short questions. If you get a 5-mark question and you've written for 2 pages, you've probably wasted quite a lot of time there because you've probably got the marks in the first half page, or maybe the first page is at the most. Conversely, taking too little time on relatively long answer questions, you generally speaking get at least 1 answer book. For my exams, you will always get 2. You should probably take that as a hint. The law of diminishing returns sadly applies to exam answers. If you write a long answer, you're going to get most of your marks in the first half, and then on a sliding scale, you can continue to work, but eventually you're just going to not be working effectively, shall we say. We don't want to see in the question repeated parafashion, which is often what you see, the question reiterated, sometimes in several different ways, without actually answering it. Waffling, that would be an example of waffling, talking not particularly on the point, shall we say. Info dumping, I guess, is an example of answering the wrong question. Okay, the importance of your own notes. This is far and away the most important section of this particular ramble, hence the big red font. I can't stress how important your own notes are. Take lecture notes and rewrite them, making sure you understand them. This is not something you can do in a week or so before the exam. You must keep lecture notes through the year, or at least start developing them as soon as you know you have a resit. If you have a resit, it's a thing. If you work hard, you'll be successful and go on to next year and complete your degree successfully. But you're going to have to start working as soon as you find out. The resits will be in September. You need to start working as soon as you find out, to give you a couple of months to make sure you're successful. Okay, I'm going to have to clip it a bit out there. You might be a bit sick of myself and other staff saying this, but the students who succeed are the ones who plan their studies in a rigorous fashion. Lectures are a foundation for your understanding. We must build on those foundations. Typical module will have around 200 hours learning associated with it. We might spend a total of 50 hours in the classroom or the lab. The other 150 hours are up to you. Put another way, during the term, for every hour spent in a timetable session, you should be looking at spending 1.5 to 2 hours working on the module. Of course, this does include time spent on assignments such as writing up essays or writing up practicals. Some modules are assessed only by in-course assessments, and here you'll be spending a lot of your time researching essays, for example. Your notes will provide you the basis for understanding the topic in many cases, but it will come as no surprise that you have to do a lot of reading around and about the topic. In this case, summarising your notes in a way that helps you understand them is an important step to developing your understanding. In the case of modules of an exam, of course, this is vital. In order to develop your understanding, you will want to use other resources, and I strongly recommend textbooks. Modules all have recommended textbooks, such as Fryan for the Human Metabolism and Endocrinology module, which students should own. There is a library, of course, which is where the physical books live, and repositories of electronic books such as eBury. Make sure you find a couple of good textbooks to support your learning for any particular area of knowledge in a module. Also relevant to the sources cited in the lectures. These were used to develop lectures, so it's worth familiarising yourself with them. Okay, we're going to have a look at a particular lecture and drill down to the detail of one of the topics. In this case, the lecture is Lecture 6 of Human Metabolism. Page 2 of lecture notes summarises the content of the lecture. It tells you we're going to focus on the first major topic of the lecture, G proteins. What are they? What do you need to understand them? The topics list makes it clear that you need to understand their morphology and role in taste. So moving on to learning outcomes. This is the stuff you need to understand. Notice the first point expands on the topic's structure, that is morphology, relates to function, and that is ultimately what we will want to understand. G protein is involved in detecting a number of classic tastes, but not sodium, which uses ion channels. So there's a little piece of information there, and when, for example, if you're writing an essay on G proteins, we would expect you to demonstrate your understanding of them. You may want to also demonstrate that you've done a little bit of reading around the topic by bringing in little relevant pieces of information like that. Okay, there should be some terms here you should be thinking about defining. We'll talk about G proteins in a bit, but first think about two keywords, morphology and taste. As you'll know from anatomy, morphology refers to the shape of a structure. It's a bit more complicated here, but shape's a good place to start. You will need to be able to describe and or draw a typical G protein. In morphological terms, you'll also be thinking about how this relates to function as well. Taste, another keyword here. This was covered in the first lecture on this topic, which will be lecture 5, and you may remember that this is one component of the multifactorial sensory modality we call flavour. It's a nice scientific language there. We're always impressed by scientific language. You will probably need to go back to the lecture and review this. It's an important lesson. Understand the details. To understand the details aspects of a topic, you do need to place them into context. Or in this case, I've done a mind map, a very simple mind map. As discussed in the previous revision video, it's up to you to find out what ways of learning suit you. Some people like mind maps, some people like writing, some people like other visual tools. For completeness, I will mention that I use the free online mind mapping tool MindMeister for these maps. Again, as previously discussed, if you develop your own maps or other text or visual tools for revision, pen and ink is much better than doing them on a computer generally. There's good evidence that the cognitive load involved in computer use distracts from learning. Okay, so morphology. Here's a pretty picture from the notes, and you may be able to figure out this is a transmembrane protein with a bunch of helical domains and some other bits and pieces. If you can, well done. I certainly needed to simplify things, and this diagram can be found a few slides on in the notes. This hopefully is clearer. You can see straight away we're dealing with a membrane, which, as membranes do, separates the inside of the cell from the surrounding medium. There are 7 helical transmembrane proteins linked by lipid chains. The helical subunits have just been drawn as simple cylinders here. Not quite correct, but fine if you label them properly. There's also 3 subunits labelled alpha, beta and gamma, and lurking nearby these subunits is a molecule of GTP. On the other side of the membrane is a ligand. Okay, so that's when we're starting to link morphology with function. This is where the where the response of the receptor starts. If you were talking about G proteins, you have to explain what happens to the 3 protein subunits, what happens to the G proteins, and the other factors that are involved. Okay, so here's the story so far, sketched out as a mind map. As well as what we know, I've added a couple of questions. We know that the exergonic hydrolysis of ATP is often coupled to endergonic processes. Is that also true of GTP here? We need to find out what its actual role is. And I've added a big red arrow to remind me that function arises from morphology, but how? It's another area that needs to be reviewed to develop your understanding of G proteins and their role in taste. Okay, that's as much as I want to say about analysing lectures and developing your own notes from them. It really is up to you how you approach this. Make sure you give plenty of time and thought, and you will be rewarded with success. Okay, don't get a bit carried away. These are examples, not necessarily what will be in the exam or the reset. You need to aim for a broad understanding of the topic and not just gamble on a few topics coming up. Okay, it's obviously difficult to talk about exam questions without including some examples, which is why we will be making the past papers available. Okay, here's an example question. Describe cellular respiration. I've highlighted describe, and there's 15 marks suggesting that you need to write a reasonably long description of the process of cellular respiration. As I always say with everything, it needs to be placed in context. And the context here is energy metabolism. So review the overall process, look at the equation, look at the, be aware that this is the oxidation of carbon atoms to carbon dioxide and reduction of oxygen to water. Think about the various pathways involved, glycolysis, the TCA, oxidative phosphorylation, electron transport. Discuss the key features. Now you can do them in a number of ways. You can write a detailed report, a detailed essay, and that's fine. I've seen people do that very good. That's a very good approach. You can draw a diagram. Okay, diagrams are good, and certainly when these exam was run this summer, a number of people did diagrams, but they didn't then develop an explanation of the diagrams. The diagram in its own won't be enough. It's a good place to hang understanding of. And as I mentioned a couple of times, it requires a substantial answer. 1 page will be nothing like enough here. Here's one where it's asking you to compare things. So it's comparing 2 types of phosphorylation. Could be a number of different examples here, depending on if it's 5 or 10 marks. Notice different synonyms can be used. If you're from overseas, particularly, and English isn't your first language, probably a good idea to think about the different synonyms. Maybe look into a thesaurus for different words that mean essentially the same as compare. Okay, in this case we look at the differences and similarities, and one similarity would be they both produce ATP. The differences, where they happen, how much ATP each one produces. If there's only 5 marks, just a simple comparison. 10 marks, we'd expect that, but also being placed in context. So the context here would be a substrate level phosphorylation, glycolysis for oxidative phosphorylation, electron transport, and oxidative phosphorylation, which are all part, as we discussed a while ago, of cellular respiration. Okay, here's one which will get you 5 marks for doing something fairly straightforward. What's a great to describe free energy? A short but complete answer, and it's only asking for an equation, but you do need to describe the bits of the equation, which is why people lose marks in that. I'll just move back. So it's delta G equals delta H minus T delta S. So you need to explain that delta G is the change in the free energy of the process. Okay, list of the components of a satiety cascade, 5 marks. It's just a list. It's not asking you to write 2 pages on satiety cascade. It's just a list. I'll put a little bit there more. You might want to sort of briefly mention context, I guess. But no, actually, you wouldn't. It's just a list, so you just want the list. Okay, describe, 10, possibly actually 15 marks this, I would suggest. A fuller answer, but still the list is a good place to start. Context in this case, we'll be looking at things like energy balance and homeostasis. Discuss the mechanisms, which were on the previous page there. It's a short-term process, so you might want to also consider, in terms of context, longer-term control mechanisms, insulin, glucagon, leptin and ghrelin, for example. Okay, describe the structure of NAD+. You can do it in pictures, you can do it in words. It's a good idea that you know its full name. In a helicopter view, this would be it's a coenzyme, which is dinucleotide, with the two nucleotides linked by phosphate group. It's adenine and nicotinamide. You need to know which is which. Talking about its role as an oxidizing agent. As we discussed a few times, you don't need to draw the whole thing out. It's just NAD plus and NADH. But it's a good idea to know which parts of the molecule has changed. Okay, so it gets oxidized, so where's it reduced? You probably want to talk a little bit about that. The case study questions, which you may sometimes say are a bit different. In this case, for the human metabolism module, you get a case study based on diet and conditions associated with diet. So read the case study information, read the questions, and then look to link the case study information with questions. Okay, good and not so good answers. We sort of said this earlier on. We don't want to just restate the question. We need to expand on it. So discuss with examples potential use of bitterness blockers in the food industry. So in the answer, I've expanded on that. I've said they are novel food ingredients and they have a potential for replacing traditional bitterness blockers such as sugar and salt. Again, as I mentioned a bit earlier on, the law of diminishing returns does apply. So make your main points early, get your marks in early. Then you can think about expanding them or coming back later. Use scientific language. Taste is a sensory modality. Sensory evaluation, not food tasting. Okay, final comments. This is the typical front page. It tells you it is 2 hours and you can do 4 questions out of 7 and each one's worth 25. You can spend about 10 minutes reading. 120 minus 10 gives you 110, so about 25 minutes per question, which you work it out is about 1 minute per mark. Bear that in mind, especially when you're doing short answer questions. Obvious stuff, people do wander around occasionally, not sure where the exam is, and also remember you do need your university cards to get in. If you've lost it and there's time, go to the library and they will print you another one. Otherwise, find a member of staff. Turn up plenty of time before the start, at least 20 minutes answer. Okay, in your answer book, clearly indicate what question you're answering. If you spread the answer around, make sure you clearly identify parts of questions. If you put a brilliant answer to question 1 in the middle of question 2, sadly you won't get any marks for it. Always start a question, so I would recommend always start a question on the new page. That's good because it also leaves some room normally. Make sure your name is in the appropriate place and your number in the appropriate place. And write as clearly as you can. I know it's quite hard actually, which is another good reason for doing revision by writing. We don't often write for 2 hours at a time. If you've got any diagrams, make them large, clear and well labelled, and as I said earlier on, expand on them in a brief discussion. And there we go, work hard and you won't need luck, and we'll see you all next year. Okay, thanks very much for listening.